Welcome to the 24th Annual Benjamin Ide Wheeler Society Lecture and Tea. And use your brain power before we start to wish for sun by the time that the lecture is over, and we're hoping that it breaks over at Alumni House. I'm Kevin Crilly. I'm the Director of Gift Planning, so I wanted to welcome you on behalf of the frontline fundraisers in the Office of Gift Planning. Melanie Kyleholtz, Amanto Marcatulli is over here, and Randy Silverman. Where are you, Randy? Oh, Randy's way up there. So welcome, all of you. We're really honored today to have Marian Diamond as our speaker, because she really embodies the mission of UC Berkeley of teaching, research, and public service. She received her BA and her MA and her PhD from UC Berkeley. And she's an emeritus professor of integrative biology, but emeritus hasn't stopped her. She will be teaching one course next semester and probably two the following semester. And she allows me to tell you, I was born in 1959, and Professor Diamond has been teaching longer than I've been alive. She is a world-renowned authority on the brain, and her pioneering research on the effects of environmental enrichment on brain development is where her research is. And her public service does not stop in this country. She is involved in a program, Enrichment in Action, in Cambodia to help children there with supplementing vitamins, teaching them computer skills and English, and helping them achieve wider social acceptance. And probably most importantly, or I shouldn't say most importantly, you can't weigh all these things, they're all important. Her iTunes videos on the UC Berkeley iTunes site has been viewed more than 2.5 million times. So, without hesitation, Professor Marian Diamond.
I can hear you whispering what I brought out. How many have seen brains before? How many have studied brains? I wish to dedicate my talk this afternoon to my mother, Rosa Wampfler, and my father, Dr. Montague Cleaves. Without their genes and efforts, I would not be receiving the honor to address you today. It feels so good to do that for my parents, for what all they did for me. So with that, let's begin. The main character this afternoon will be the human brain. How many of you have thought about your brains this morning? Quite a few of you. How many of you have thought about your brains thinking about their brains? Doesn't that literally blow your mind? What you carry in the top of your heads? This was a former student. Have you ever asked yourself, how can little cells think when they're too small for the human eye to see without additional magnification? We should stop right here as you marvel about this 3-pound mass. It only weighs 3 pounds. That you carry in your heads. Just allow a few minutes for you to truly appreciate this brain. Do you feel it? Anybody feel their brain up there? Do you hear it? Do you see it? Do you smell it? The answer is none of the above, but you know it's there, and it's responsible for all your integrative functions and consequent behavior. All of them. It only weighs 3 pounds, and yet I can hold it in one hand when it previously had the potential to conceive of a universe 1 billion light years across. Little cells. Who built the Golden Gate Bridge? Who built your Campanile? Do you ever give it the credit it's due? Once a TV producer came to my office and thought it would be interesting to have a brain scan. Consequently, he did, and learned that his whole left frontal lobe was missing. Do you really know what's inside your skull? You ever thought about it? Does anybody know? Nobody's had a brain scan? Was it positive? That's it. I already gave this to my students. In my short talk, I'll attempt to teach you some basic facts about brains, as requested by some of your members. They said they just didn't know about brains and they'd like to learn. So I'm going to be very basic. Now, I've got to learn how to deal with this slide. And take these off. We used to fix them in formaldehyde. And all these decades, we worked over formaldehyde, and then they decided that formaldehyde was carcinogenic. So now we fix them in formaldehyde and then transfer it to alcohol. And then I tell the students, and yes, alcohol can preserve the brain too. So you have dual messages to offer here. All right, slide number 1. Let's see how we go. I press forward and nothing. There we go. Thank you. Now we're going to start very primitively. We're going to start in the embryo when the brain is starting to form from a tube. It's called a neural tube. Nerve cells form the tube. And we have a channel down the inside, and we're going to see what that turns into as it develops. We want 3 divisions. There's a hindbrain, a midbrain, and a forebrain. I'm sorry why I'm slow with this, because the lights up there are shining down in my eyes and I can't really see it as clearly as you possibly can. So if I do something, you tell me. We're a class, we're a family, we can talk, all right? It's okay. Can they, I don't know. Can these lights overhead be dimmed? Not at the moment. So, slide number 2. That tube that we saw before with the hindbrain, forebrain, midbrain will form our human brain here. The rest of the tube will form your spinal cord. And the spinal cord is sending out information through spinal nerves. You have 30 pairs of spinal nerves going to your body. And the information that they bring in can travel up to the brain, carry out processing within the brain, and then come down again. Your spinal cord's like a freeway. Information coming all off, and then off ramps, on ramps to come in to keep traveling. Many functions of the brain, then, are correlated with functions of the cord. So my first question to you, I'm going to be asking you questions. Do you think it possible to have a complete brain transplant from what you've just learned? No, of course not. People say they want a brain transplant. You cut off here, you cut off all the signaling that's going here. Some of my students who've become neurosurgeons will say they try little pieces of brain transplanting here, but they destroy so much putting it in that they gave that up. I don't know where brain transplanting with small areas in the brain is right now. Now, how do we make a brain? I hope you can see this. This is a slide, a histological slide showing nerve cells. Brains are made of only two kinds of cells, nerve cells and glial cells. Glia in Latin means glue. They thought that the little glial cells glued the nerve cells together. During this lecture, you'll find out how dynamic glial cells are. Phenomenally dynamic. They have a symbiotic relationship with the neuron. So neurons need glial cells, and glial cells need neurons to carry out their functions. Most brain tumors are gliomas, not neuromas. Have you heard of the glioblastoma? Multiforme, it's still the most deadly brain tumor, made of glial cells, and usually less than a year to live after diagnosis. Have you known anyone with a glioblastoma multiforme? You? My brother had one. Once it's diagnosed, there's roughly a year at most to live. This brain slice shows no tumor. This is a normal, healthy brain. Does anybody have any idea whose brain it is? It's Einstein's. I had the courage to be one of the first to get a slice of Einstein's brain. And we made a study from his brain with 11 other male brains. And it turned out that we found that, I've got notes because otherwise I get off a tangent and we don't get finished. So I'm trying to keep on tangent with you. I think we'll come back to glial cells later, but these are Einstein's glial cells, and these are the nerve cells. Aren't they clear and well preserved? They're beautiful. Most histology that you get with these are not so clear. Now let's get... Nerve cells have branches. And I used my hand when I was director of the Lawrence Hall of Science. I had little kids, so I was doing a lot of kid teaching. A nerve cell starts as a circle. And as it gets stimulated, it'll send out its processes. These processes are called dendrites. They receive the information from other nerve cells. This is the cell body, and then transmit it through an axon to other parts in the nervous system. So you're seeing here a real nerve cell, body, branches, and an axon to convey the information away. There's so much to tell you that I have to skip to, but these things are so exciting when you start studying brain cells. Now let's see. We decided that we would coin the phrase, use it or lose it, with regarding the brain. And we did this long before it's so popular with everything now. Because when we have a brain cell that's round and it's formed dendrites, with use you grow dendrites. With disuse, you lose them. And we can show in 4 days, we can have a significant loss if nothing's coming in. So you want to use your brains, not so that you get them tired, but with reasonable input for use it or lose it regarding the brains. That started in our lab. Now, axons can be various lengths. I showed you the spinal cord. You came clear down to the base of the spinal cord. There you're going to have nerve cells that send their axons clear to your toes. How long is your leg? 4 feet, 5 feet? That can be a single axon. Move your big toe. Anybody can't move a big toe? Want to be sure the process is there. Your axon can go clear down there. Remarkable cells. They can be at least 3 to 4 feet long. Now let us return to more detail for each division. Let's look at the hindbrain. This is the picture we saw to begin with. We had the tube with hindbrain, midbrain, forebrain developing. Here we are further along. The red represents hindbrain. On the foreside, we have what's called the medulla. That's essential to your life, that 1 inch of brain tissue. This is spinal cord going on. This is the beginning of your brain. And this is the medulla. And the medulla, we want to say this deals with heartbeat and with respiration. So you know why it's important. It's essential to your life. The cerebellum, back here, deals with many other functions, but primarily balance and coordination. Would you like to test your cerebellums to see if they're working while you're sitting there? It's easy. Take your right hand, put it out. Don't hit your neighbor. To put it as far as you can. Have an index finger pointing too. Now close your eyes and slowly bring your index finger to touch the tip of your nose. Eyes closed. Slowly. You're too fast. You did it. You got there. Did anybody miss? Do it again. No, seriously. This is a clinical test for testing cerebellums. Your eyes closed. Eyes closed. Eyes closed. If you really miss, you should see your neurologist because it's a very simple test. Many of you, did you know Bob Karplus? He was a UCB physics professor. He had a stroke while he was running in the Berkeley hills, and as long as he lived, when he came to my office, he never could get back to his nose. But do it with your left hand. Be sure they're both working. While teaching in Finland one summer, I found a woman in the audience who couldn't touch her nose. I said, go see a neurologist. She had a brain tumor, had it removed immediately. Simple test to give you what's going on inside your brain. And as I say, I mention these. I also repeat lots because you learn with repetition. So you'll say, well, I already told you that. But things that are important, I'll tell you again. And so I mention this test because sometimes it's important to learn just a simple test can be very useful for clinical diagnosis. Now let's go to the next slide. It's a little darker. This is a section through your midbrain. I'm going to take it back. Here was our hindbrain, our medulla, and our cerebellum. This little yellow area in here is your midbrain. It's the smallest division in your brain of hindbrain, midbrain, forebrain, but it's packed with function. So let's take a slice through it and see what it looks like. If you want me to repeat anything or to speak louder or anything, please talk. I consider this a family. We just here to share. I'm sorry? This is yellow. This is blue. This is going to be forebrain. This is midbrain. This in between, that's your thalamic. I'm not going to go into that one, but it would be associated with forebrain. All right? Thanks for your question. Let's see now, we want to get midbrain again. Now we've taken a section. There are many different ways to stain brain sections to bring out different characteristics. 3 terms I'm going to use here. I'm going to use colliculi, I'm going to use substantia nigra, and I'm going to use peduncles. Let's go back to colliculi. Little bumps. Colliculi are little hills. These are superior colliculi. They do with visual reflexes. Anybody play tennis? You have to be very alert with visual reflexes getting that ball back and forth. Midbrain, superior colliculi. Down the brainstem from the superior are the inferior colliculi for auditory reflexes. So these are both midbrain structures dealing with vision and with hearing. Now, if we come back here, this big one here is called substantia nigra. What does that sound like to you? Black substance. You say, but it's white. It's the preparation. In the fresh brain, this area, your substantia nigra, is black, very black. And how many of you have heard of the neurotransmitter dopamine? Quite a few of you. Dopamine is produced in the substantia nigra. And we can test to see your dopamine very crudely. Put out your hands. Put both hands side by side. And do you have calm fingers? Very nice calm in the front rows. I can't see much beyond, but you could tell me if you have any slight tremor beyond. Beginning of Parkinson's. Because here I see the second row is going like this all of a sudden. Well, we might as well laugh. We have the choice, so we can do it either way. Right, thank you for demonstrating. But I want you to know that every millimeter in the brain does a different function. It is so miraculous, this structure you take for granted up there. So we've seen now the substantia nigra, that if it reduces its dopamine, you're going to have beginning of Parkinson's. My garage mechanic had a Parkinsonian tremor, and I'd come to get my car fixed, and his hands were going like this. Did I worry? No, because once I told him what was wrong and he picks up a wrench, he loses the tremor. But there's another kind of tremor, which is called intention tremor. And you're perfectly fine while you're sitting, but when you go to do something, you get this. Sure, you can check your dopamine, find out what's happening. I don't know if people injecting it, no. I'm not pretend to know all of this. You have lots of questions that are out here that I haven't studied. I've been here 50 years and I've studied a lot, but there's so many with the brain. Sorry. But write them down and give them to me, and I'll try to find the people that can answer them for you. So my question to you is, would you want a brain surgeon with a tremor at rest, when it's rest like this, or one which is an intention tremor? Which one do you want? At rest, sure, so I feel safe with my mechanic. And when he's shaking, that's more significant than if he were calm and then started to shake. Cerebral peduncles. They're large here. What are they? These big black things here. These are fiber tracts that are coming way up from the big hemispheres here. These are your hemispheres. Those fibers are coming down and they're collecting as they go through the midbrain here. So if I injure those fibers here, I will have paralysis on the opposite side of my body. So when you study the brain, there's so much that's going on. Every millimeter is different. And they cross. They're coming down one half of the brain, and then they cross to the opposite side. And everybody asks, well, why do they cross? Not many people know. But they do, so it'll be the opposite side if I cut them here that will lose function. So those are just some of the functions going on in your midbrain. While I have the midbrain slide, does anybody remember what we call this opening in the neural tube? That was a central canal. Here it is down in the midbrain. We have that central canal going through the whole brain stem. And what does it carry? It carries cerebral spinal fluid. You're forming 500 cc's of cerebral spinal fluid every 24 hours. Did you know that? You're forming it inside your brain. Where's it go? In the hindbrain, it comes out and goes around the brain and gets back into your cardiovascular system. So it's circulating all the time. What happens if you get a tumor and block it? If you're young, you get hydrocephalus. Your brain will just swell because the skull will swell too. How do they treat that today? It's more common than you think. With a shunt, right. They take a shunt, which is just a tubing, and they put it into your midbrain. If you're blocked above the midbrain, you put it in there, and the shunt will carry the cerebral spinal fluid under the skin of your scalp, down your neck, down your chest, to your abdominal cavity. And you'll be releasing CSF into your abdominal cavity. We had a professor here. He fell in his backyard, and he blocked the tissue around the Here's my channel. It's got CSF. But he blocked with tissue with this so it couldn't continue to flow into the hindbrain. And so, they put a shunt in, and he loved to come to my class and have the students feel his shunt. Because you could palpate it all the way down. But you got the principle of what can be done when something goes wrong with this brain. So let's see. So how do they know how large the ventricles are? The next slide, after death. They can make models of the ventricles. I showed you a slide that cut right through this little cerebral aqueduct or channel here. But all of this is ventricular. What they do after death will inject either a metal or a plastic, and it will fill the ventricles. So these were all channels with CSF flow. And going out here, down here into the hindbrain, and then it'll go back around the brain and come back here and get into the vascular system. So this is what your ventricles look like without any brain tissue that's been dissolved away from it. And now we come to the forebrain. This is the picture you usually see in your medical texts. Here's my medulla way back here. Here's my spinal cord, little old friends for you now. Here's the cerebellum. How do you test for your cerebellum? You remember, right? Good for you. But 85% of your brain, it's these large hemispheres, your cerebral hemispheres. And you see how folded they are. How big would they be if they weren't folded? Why is it essential that the big cerebral hemispheres fold? About 2.5 square feet if they were stretched without folds. Why can't you have a brain that's 2.5 square feet? One reason. Why? Pardon? One reason. I don't hear it. It wouldn't fit your skull. Why is it important to fit in the skull? Pardon? No. Come on. Pardon? How many of you have delivered a baby? Would you like one that was 2.5 square feet? There's only one reason it's got to be that size. It's barely making it through the female pelvis as it is. But for it to get there, that brain has, as it grows, has to fold. In my little rats, theirs have yet to have a fold. You get them in cats, dogs, monkeys, and men, but you don't in women, but you don't get them in those that are still growing. But this has folded, so we have to learn all the folds. They all have names, they all have functions. We're going to look at the lobes of the cerebral hemispheres, the most highly evolved mass on our Earth. And here, if we cut through here and here, this will be the occipital lobe. What happens in the occipital lobe? Just one word for you. Vision. But you say, this is front, eyes are out here. Why do we have vision back here? Who knows why? But we do. So the impulses have to come all the way back to your occipital lobe, your visual cortex. You have a large representation for vision. This is what we call the lateral aspect. The lateral aspect up here, this is a different lobe. All of this. Anybody know the name of it? Temporal lobe. What's it got to do with time? You ask any neurologist, they don't know. We don't know, but you guess. So where's the first area in your body that you can see the passage of time? The temples. What's happening there as you get aged? First sign of graying. Does anybody get the first sign of graying up here? No? All right, first sign of graying right here at the side, temporal lobe. And right underneath this fold has your auditory cortex. I'm sorry? I can see it. There it is. It really disappeared, or you're teasing me? That's okay. I don't mind being teased if I can't see it. You've got it back again. No, thanks. There it is. Now that's your auditory part of your cortex. So you know where it is now? You could feel underneath. So for you to be listening to me, it's firing. For you to be seeing this, sometimes when the laser's working, your visual is here. So what other ones do we want to pick up here? So which do you think? Question. How much of our education deals with visual input and how much with auditory input? Right there. Yeah. There's auditory. All right, let's see on our next one. More visual. How much percentage did you say? 80% visual. Something to challenge, that's what it's all about. Now let's go deep in the temporal lobe. What do you think you're going to find when you go away from the surface? We've cut all the cortical tissue away, and we have a structure like this. Does anybody know what it's called? This is the hippocampus. Horse field. Does it look like a racetrack? Evidently, the early anatomists thought so. And why is this so important? Why do most people like to learn about the hippocampus? Because here, recent memory is first processed to be transferred later into long-term memory out in the cortex out here. But recent memory processing, how did they learn that? Because there was a man in Canada who had epileptic seizures, and the neurosurgeon decided if he took out both hippocampi, right and left, he would reduce those seizures. So he did. What happened? the man could no longer process recent memory. No recent memory since then. He just died a few years ago. He had long-term memory before, but he couldn't because these were not. So these are very important for your recent memory to process, to get it out to your cortex.
Did it cure the epilepsy?
I'm sorry?
Did it cure the epilepsy?
Cured the epilepsy, yes. Which choice would you want? Who knows? All right, another thing that I like about the hippocampus, it deals with visual spatial acuity. And I personally appreciate this, developing this sensory modality, because I do a lot of traveling and I leave my car in the airport. But if I walk away from my car towards the terminal and turn around and get the ambience of the environment around my car, my visual spatial function, when I come out of the airport later, I can go right to my car. How many of you do that? Quite a few, right? But you can see it's your hippocampus, which allows that to be so clear. You save so much time in an airport. Then the parietal cortex. We're back to, here's our medulla. Here's our cerebellum. Here's our cord. Here's our temporal lobe, our occipital lobe. And the parietal cortex, parietal is wall, so it's the side here. And I want to take one of these. You have a central sulcus that comes down, and the fold beyond that is primary sensory cortex. Well, you said we already did with sensory, visual, and auditory. But what's this one doing? All your other sensations. As we come into, which ones do we want to take? Touch, pressure, temperature, kinesthetic sense. Do you know what kinesthetic sense? Your position in space. Terribly important. But all of those are coming in to your postcentral gyrus. Your precentral gyrus is motor. For me to be moving this pointer now, my precentral gyrus right about here is firing. And then this is the prefrontal lobe. And it deals with higher cognitive processing, with judgment, initiative. It's the most highly evolved part. We counted the glial cells there, and it has more glial cells than other parts of the cerebral cortex. This is where we've been evolving most in our brain, is pushing this out. If you look at primitive man's brain, it was this way, and now our frontal lobe's going this way. And my question is, with the stress we're going through now, is that going to send it back? Or will our enrichment bring it forward still? Who knows? Stress does reduce cortical dimensions. We've proven that in the lab too. So let's see the next one to see. These are how the sensory and motor representations are on your precentral gyrus and postcentral gyrus. Most of it's an upside down representation of the brain. So they're getting inputs from your toes way down here, but come around. Look at how small in your legs. Very little innervation, but look at the hand. Tremendous. How many of you are violin players? My son has played in the Berkeley Symphony for all of his life, and I watched the rapidity. I'm sure his representation on his cortex is phenomenal, as are all violin players, but look at it. But then the thing that we don't understand is, why is the face right side up? Everything else was upside down. And look at the mouth. Huge mouth. Have you ever tried to talk without moving your tongue or your mouth? It's no possibility. Extremely rich in sensations and motor skills. Let's see, which ones do we want to bring in here? I'm skipping lots and trying to get those which I think you will find most useful.
Does pain register in individual sections? Just right there, or is there a central area?
No, pain will be in all areas, depending on which area. If it's going to be mouth, it's going to be your throat in here. Big toe up here. All right, let's just ask the question then, what are some of the functions of glial cells? This is why we worked on Einstein's brain to find out his glial cell components versus the normal male. And we found certain areas where he had significantly more glial cells than the average male. And so people have always asked, well, why did you do it and what happens with glial cells? So I had a professor friend, Gerhard von Bonin. Does anybody who studied the brain know Professor von Bonin? Well, he came to the lab and he suggested that the parietal lobe was more highly evolved than the frontal lobe, and that the parietal lobe pushed the frontal lobe forward. And so as you increase the evolutionary scale, the glial cells increase in number. So I reasoned that the part of the cortex with the greater number of glial cells would be more highly evolved. It turned out that the frontal lobe did have more glial cells per neuron. I also found that Einstein's brain did have more glial cells per neuron in the inferior parietal lobe. You know where that is now. Than the average male. Now, what are some of the functions of glial cells? Why do you want them? So I looked and found 6 more functions to give you. They play an active role in establishing and maintaining fundamental patterns of neuron circuits. We said all these fibers have basic patterns. The glial cells help determine those patterns. They produce growth factors, playing a key role in regeneration and plasticity. We've taken rats and made lesions in their brains, put some in enriched environments, some in impoverished environments, and we can show where the lesions were. We have grown new dendrites. Excuse me. But also, what was new with these lesions, if we place them in the brain, not only did the brain tissue around the lesion grow back, but the adjacent brain material grew more dendrites. We call it compensative hypertrophy. They try to make up for the dendrites that had been lost with the lesion. Tremendous effort. When the Koreans found this, they came over, they got every bit of our data to see how to get lesion brains to grow and find out what we were doing in the laboratory with our experiments. Now, some play an active role in the formation of myelin. What does myelin do? It speeds the rate of conduction. These pathways that we've been talking about, they're not all traveling at the same speed. Some are very fast, some are slow. The ones that have more myelin, they go faster. You've heard of demyelinating diseases. You erode the myelin from the fibers. What are the most common demyelinating diseases you hear of? Multiple sclerosis and amyotrophic lateral sclerosis. That's Lou Gehrig's disease. Both of these are losing the myelin. What is myelin? It's a lipoprotein. So it's fat and protein. What color is the fat on your beef when you buy it at the market? White. So it makes up your white matter. It's the white covering of your nerve fibers. The more myelin you have, the more rapid the impulse. You get these demyelinating diseases, you lose these functions. Some glial cells respond to rapid repair of myelin in demyelinating diseases such as multiple sclerosis and ALS. Some glial cells play a crucial role in immunological responses to various infections and toxics. It's hardly anything you think about in the brain that these glial cells aren't participating now. And before, where did they get their name? What does glia mean? Glue. That's what the early anatomists thought. See how far we've come? They've come to really gaining stature with knowledge about the brain, but it's so hard to get started on all these. So with these definitives, we can appreciate how important glial cells are to the well-being of nerve cells. Now, what factors did we use in our labs and consider essential for enriching brains? We find 5 of them that we've tested in the lab that are very important. First, diet. Next, exercise. Next, challenge. Next, newness. And last, love. You're beginning to hear these on the radio, all but love now. If anybody tells you that love is good for the brain, we'll give you an experiment that will show it. But diet, importance of protein. While teaching in Africa in 1988, we found that many women would not eat protein when they were pregnant because they delivered too large a baby. They watched their friends die in childbirth, cut out the protein, have a little baby. Came back to Berkeley, worked on these, and with a graduate student in nutrition, Ariana Karugi, and found if you cut the protein during development, sure, you get a small baby. But do you have dendrites that are out there? No, you have them here. So you need the protein to form healthy nerve cells. Well, with glial cells, with all the things that I mentioned, exercise, diet, just keep a healthy brain. And use, that's why we put in challenge. Our rats, we would challenge them. Pardon? Getting time for questions. Okay, let's see what we've got. I just want to be sure that I get the highlights set. But anyhow, protein, terribly important to form healthy nerve cells. And fruits and vegetables, everything that you're learning now. When we were in New Orleans, the people were getting for breakfast, they'd have 3 different fruits. When we saw that, we changed our diets too. For breakfast, we have a banana, an orange, and berries or something. But fruits are exceedingly useful for healthy brains. And enriched environments, don't just sit and pass out the last 40 years of your life. Plan, do things. I mean, we show the effects of impoverishment, it's tragic. Join things, do things, eat well, but eat well is number 1, because if you don't build the dendrites, you don't have anything to respond to in the environment. Now, I'll take just one more thing. With all of this, we went to the most impoverished children we can find in the world. They were in Cambodia, and they lived in the forest, had no running water, had no electricity, had no toilets, had nothing except these monks who would bring some food. We gave them vitamins, we taught them English, and we taught them computers. I now get emails in English after 10 years in Cambodia. So it can be done. The brain is plastic, so use it or lose it. I'm cutting short because I'm told to. Thank you.
Microphones out there so they can.
I see. Right. Thank you, because I was going to say if nobody has any questions, I'm going to bring in love. We were raising our rats and seeing how old they could get, and we were beginning to lose them at 600 days. 600 days roughly is equivalent to 60-year-old people. all right? We were losing them. Why? Those days I went out and visited lots of old people's homes and said, what's missing here? They had good food, they had activities, they had everything. But I said, where was the TLC? Where's the tender, loving care? They would come out of their rooms, go watch the television or go to eat, but nobody was sharing anything with love. So we decided to pet our rats while their cages were being cleaned. So we'd hold them against our lab coats. Yes?
If a person has Alzheimer's or Parkinson's, is their brain the same?
I'll come back to that one. I'm in the middle of this one. May I finish it and then come back? I'm in a positive one right now. I'm not laughing at you. I'm just saying that I don't want to cut her question, which was about love. And so we gave love to our animals. Long story short, we got those up to 904 days of age. And we asked around. I mean, the German rats were getting up to maybe 800, but nobody had gotten them up to 900. And we were very successful with that. So we say now that include in my 5 priorities, we've said first you have to have diet, you have to have exercise, challenge. You don't do the same old crossword puzzles all the time. You get new challenges. And you need, I'm going to skip and get to love. And I don't mean sexual love. I mean love above like. You have lots of friends you like, and you have some friends you love. But tell them that you love them. And you feel so much better. But beginning to bring the word love into the conversation, but as soon as you take the sexual component, everybody relaxes. Well, it's reasonable that it should be there for the reasons it's there. But when we get older, to tell somebody you love them, it feels awfully good. And that's why we put it in, because that's what we saw was missing. And people couldn't believe we got our rats up to 900 days. Now, our gentleman here, would you repeat your question? It was Alzheimer's, and it was...
If a person has Parkinson's or Alzheimer's, is their brain the same?
No. As I gave you, Parkinson's is a depletion of dopamine. Alzheimer's can be more extensive. It's not located just in one region, as I gave you with Parkinson's, which was dealing with the dopamine in the basal ganglia. All right?
Could you tell us a little more about, I think you called it intentional tremors, and the relationship of that to the tremors one gets from Parkinson's?
Well, Parkinson's is a tremor at rest. When I'm sitting here like this, I'm not doing anything. I just have my tremor. But when I go to do something and get a tremor, that's intention tremor, and the cerebellum is feeding in.
Is the attention tremor also from the lack of dopamine, or is it from other
I don't know. I didn't find anything on that. I'm sure every question you ask, you can go to Google and you get answers. Sure. But it's true. I mean, the field has blossomed so much, and you see how complex it is. And we take certain parts of it and really do well with that, and all the rest is out there yet to be done. So if students ask you what they should be studying, brains, 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 maybe someday we'll get rid of the negatives that the brains do all over this world. Understand it. I'm trying to inject love now, and it's okay to love. It feels good.
Dr. Diamond? Hi.
Yes.
I want to give you some love. I had you in 1974 as I was one of your students.
Oh,
And truly one of my finest teachers. And so I love you.
Well, thank you. I love you too. That's very nice of you. Right.
I have
But see how much better you feel when you all of a sudden
I
add
do.
that to your vocabulary.
I
You just sort of relax. They understand.
Yeah, I just wanted to ask one more question, though.
Yes.
I have a strong interest in autism, work with students with autism. And I'm wondering, the brain, if those little glial that you talked about, they're affecting the communication areas of the brain for the child, and sensory is what my experience has been. So
The glia are everywhere.
they're everywhere.
Yes.
What does a child with autism, what does their brain look like?
I wish I knew. I haven't studied autism. I heard something last night about it, but it wasn't focusing with what I was doing, and so I don't have it for you.
Could that be one of your new challenges, number 3?
There's so much. That's what the problem is, to sift out what's the most important. And I felt love was the most important thing I could talk about here today with you, because, boy, it feels good. It's okay to love.
Is it true that the retina is actually outpocketing of cerebral
Yes.
cortex, where some people have thought that there's a thinking process going
Good
on?
question. Women are from Venus and men are from Mars. Is there any difference? Yes, we've shown sex differences in the brains of our rats. We measure their cerebral cortex. The reason we work with rats, one of them, I mean, it's a mammal, so it's in our class, but its cerebral cortex has yet to fold. So it's very smooth, very easy to make microscopic measurements to find out how it grows. Once it begins to fold, it's difficult to get common ground between the two. We find with the male, his right hemisphere is thicker than his left. And we find that at every age except very old age when he loses that right. The female has both right and left the same. And people keep saying, well, what's the meaning of it? I say I can only speculate, because it takes another lifetime or more to go in and figure out the true meaning of why they have right and left. But I said, just talking off the top of my head, I would say, what's the major function of females in the phylogenetic tree? It's to have young and take care of them. She has to be ready to go in all directions. So if she were lateralized, she couldn't do it. It's down here. So the information can go back and forth very readily. And some husbands say, when you tell them that, they say, well, my wife can just get everything, but she doesn't do anything in depth. Males have the right thicker. And in the phylogenetic tree, what is their important job? It's to find a female and defend territory as you go down to lower classes. And so those are fairly focused functions. Who knows? If you study this, you gather many more things to go in. But yes, there are, when you have the genetic disposition fairly equal in our animals in the colony, male and female differences. But we know with the human race, they're male males, they're female males, they're male females, they're all mixtures of us.
One last question. Microphone? Do you want to ask your question again?
Well, the question was, is the retina an outpocketing in the embryo of the cerebral cortex that led some people to believe that you might be able to have a thinking process with the retina, because
Can you understand that?
Can't understand.
I can't. Sorry, I can't understand it. Afterwards, I'll catch you, because it's blurred coming over the microphone. Sorry. At least, pardon? Pardon? Loss of memory? Not working at it. It takes work to keep it active. You can't just sit around and say it's going to be the same. I showed you, you've got to grow those dendrites, keep them out there. 4 days we can show a difference in them. I mean, it's hard, Before we had essential things we had to do in our lives. Now we've learned how to do all those essentials, and so they're routine. But we need new stimulation. But I figured if we could get this base and all love each other and then get some stimulation, we'd just have a much better society. Thank you.
It's very difficult to crack the 56 years of teaching into 1 hour, so we thank you, Professor Diamond.
You're welcome. And the brain is here, and it is a real brain, and if anybody wants to come up and look at it, don't do like little children do. They poke at it, they wonder what's inside. And so these are precious. I've had it for 25 years to take to the schools, but you can see your brain inside your skull, it's soft as Jell-O. This has been fixed and preserved.
You might want to look at it, maybe not right now when you're getting ready to eat, but I don't know. So please join us over at Alumni House for the tea, and we're going to stimulate different parts of your brain once we get over there.
Good.
Thank you.
Thank you very much. Thank you.